Okay, this video is going to dive into some of the fundamental theories of perception. We'll start by asking a pretty basic question. How do we recognize the things we see? And there are various ways the system might work. So for a long time, there's been a debate about how our brain does this. For example, one early theory was called template theory. The idea here is that we've got multiple templates, basically kind of prototypes of things, and those are stored in memory from past experience. So when you ask, how do we recognize what we're seeing? The answer would be that our brain compares what's hitting our eyes right now to those templates that are in our memory until it finds a match. Like, this is the incoming stimulus. What am I seeing? For my brain to recognize that, there's going to be a search through my memory until it runs into like number 2 here, at which point we know we're seeing a cat. It's a thing I've seen before. It matches the template. That's a cat. I've recognized it. But this theory is pretty naive on its surface because it doesn't explain how we deal with things like imperfect matches. Like, we can see a depiction of a cat that we've definitely never seen before and still recognize, yeah, that's a cat, right? That fits the concept of a cat, even though we couldn't possibly have that exact template stored in the brain. So if we did this matching search, going through all the things we've seen before, it wouldn't run up against any match. It would say no match, but we're able to see a cat. So template theory can't really account for the flexibility of our pattern recognition system. Think of it kind of like the CAPTCHAs you see online a lot to check if you're human. We can recognize letters of the alphabet even when they're distorted in ways we've literally never seen before and didn't memorize when we were learning the alphabet. So we need a more nuanced theory to understand how we're able to recognize something even when we've never seen that exact thing before. A sort of theory of concepts, but for visual perceiving. And the most promising way to explain at least the basics of what's going on comes from feature matching theory. So feature matching theory states that we recognize objects on the basis of a small number of basic characteristics or features that our brain, so I guess you could say what our brain is doing behind the scenes before I perceive a cat or a car or a letter H is detecting specific elements and assembling those smaller pieces, those simpler features, into more complex forms, more complex combinations of simple features. Like, what is a capital letter A other than 2 lines tilted at a particular angle and a shorter horizontal line connecting them halfway down? Like, those features are what make up a capital A and what make that capital A different from a capital F or a capital H or a capital T. So let's talk about kind of a model for how feature matching theory might work. In fact, this model I'm going to show you was created by a guy named Selfridge back in the 50s, and it was one of the first computational models in pattern recognition. The whole idea of his model is that the system recognizes and processes the individual parts before we perceive the whole. So this is a bottom-up theory of perception. He called his model the pandemonium model. And pandemonium just means like an uproar or a confusion of noise and shouting. And the word roots translate actually as every little spirit or every little demon. But we're not talking about actual spirits or demons, just metaphorical steps in a process. What we're really talking about here is a neural network. And we're talking about what neurons are doing at earlier or later steps of information processing. But the metaphor of something like demons might kind of help it click. So in his model, what he came up with back in the 50s, we've got the input coming in from our what he called image demon, meaning the neurons in the eye and kind of the earliest stages of the visual cortex that are just comparing the input to known basic features like lines and angles. Then at the next step, each what we'll call feature demon, you can think of this as the next step in neural processing. Each feature demon's job is just to shout if they detect the feature that they correspond to or the one they're looking for. Like, one demon is looking out for vertical lines, another is looking only for horizontal lines, another is looking only for oblique lines, another only for right angles or curves or whatever. Depending on how many of each feature we get, in the initial image, these feature demons will shout quieter or shout louder to the demons at the next level over. And by shout, by the way, we're just talking metaphorically. We mean neurons firing and sending a signal to the neurons at the next layer of processing. So the next layer we call is the cognitive demons in the green section here. They're just listening to the shouting from the feature demons, looking for their particular combination. Like the cognitive demon for capital letter A is looking for 2 oblique lines and 1 horizontal line. If it gets that, it gets really excited. The cognitive demons for letter O or for letter Q are both looking for a continuous curve. But the letter Q also needs an oblique line to go with the continuous curve. Meanwhile, letter T needs to hear the feature demons shout out like a single vertical line and a single horizontal line to get him fully excited. So you can think of each cognitive demon as shouting louder the more of a match there is for the thing it's excited about or the thing it cares about. So the more it hears the things it's listening for, the like elements that it's tuned to, the louder it shouts, saying, pick me, pick me, pick me, it's me, that's what I'm seeing. So like here, for example, in this one we're looking at, the D and the P, they do have some of the features of the incoming image, right? They have a partial match, a pretty decent match. So they're shouting out somewhat at least, but they're not shouting out nearly as much as R, because R has more of a match. It's getting more of a perfect match from those previous feature demons, right? So then we go to the final layer, the last layer here on the right. We've got the decision demon, which is just listening for the loudest shout from the cognitive demons, like listening for one combination demon at the green level, like, was this the best fit for the image that came in? We then perceive whatever that decision demon is settled on, and that's what we see. That's our perception. So note that this model, it works better than template matching, because even though all these different ways of writing R that I've shown here, even though those are all different, right? Even if I've never seen them before, they do have the same basic pattern of the simple features, right? It's closer to R than to any other letter in our neural architecture. So this system, this whole pandemonium model, it has a lot of flexibility to it. It doesn't have to be perfect. We're just looking for the thing that's closest to matching things we've seen before as a combination of features. So feature matching theory can kind of account for our ability to recognize things in a wide variety of circumstances and variations, and even to recognize novel versions of the kind of thing we've seen before. Here's, by the way, just another drawing of the same thing, just kind of hammer it home. You can think of the guy laying on his back over on the left there. That's our retina. The stuff going downward from him into the cauldron on the left there, those are like all the wires of the optic nerve going out from the eye to the cerebral cortex, right, to the occipital lobe where the signal is processed, just to look for basic features like a straight line or an edge. And then for slightly more complex combinations of those features, like two edges making an angle or something like that, maybe even more complex combinations of those. And by the way, this feature matching theory for how we might recognize things, it has a lot going for it. Like even with just a few simple features, we can distinguish every single letter in the alphabet, as you can see from the combinations here, by an old researcher named James Gibson. As for evidence supporting the idea that our brain actually does use some sort of feature matching theory, like yes, this is one way to explain things, but do we have any evidence that's how the brain actually does it? Yeah. There are lots of experiments that support it. So first we'll do a purely behavioral kind of cognitive experiment, and then we'll talk about some neural stuff. So to take one example, we humans, we all show a sort of perceptual confusion between letters that share many features. So in a study, what we might do is put people in front of a computer, and we're going to very, very briefly flash two letters on the screen simultaneously. So they're just staring at a screen, they're just waiting, and then two letters flash up just for a fraction of a second. And all the participant has to do is decide if those were the same letters, the two letters that came up, or different letters, okay? So I'll give you a feel. For example, if you were the participant, we'd have you just stare at the red fixation point here, and then really briefly flash some letters, just like this, in 3, 2, 1. Okay, were those the same or different? And then we do another trial like this in 3, 2, 1. And again, I'd just ask, are those the same or different, right? You'd hit a key on the keyboard, or you'd say out loud, same or different. Let's do another trial now. 3, 2, 1. And same or different. And finally, one more example. 3, 2, 1. Were those the same or different? And you probably got all of those right if you were looking. They're first we saw G and M, and then P and R. On the second set, we saw D and O, and then a U and an M. And in this case, by the way, just for this little demo I did, everything I flashed was different letters. But of course, in the real experiment, we might mix among that a bunch of trials where we flashed like two of the same letter, like two B's or two T's. And of course, we would counterbalance that out or randomize which side each letter showed up on, things like that in a real experiment. Also in the real version, the letters flash faster than what we saw here in the demo. So actually, most people don't get every question right. But the key here is that in some of our trials, the letters don't share many features. Like G and M don't have a lot of features in common, or U and M don't have a lot of features in common. Whereas in other trials, they share a lot of features, like P and R actually have a lot of features in common. In fact, there's a P embedded in the R. We just have the extra little loop going down here. Or D and O do have a lot in common, right? There's a lot of a curved line. It's an enclosed space. So some of the pairs we show common features, some of them not as much. And when we analyze people's pattern of errors, like when they get it wrong, when they mistakenly think they saw two of the same letter, it's usually for letters that share more features. Likewise, we can analyze the reaction time. And here we also find a difference. So how quickly people can answer that what they saw was the same or different. When we're just looking at those different trials, right? So we look at some different trials where things share a lot of features. Like this. And some of the different trials where things don't share a lot of features. And they're not all the same. People say different, but they say different slower for like the PR comparison, or they say different slower for like the DO comparison. It takes them longer to say those were different letters, even when they get it right. On average, actually, it's an extra 1/10 of a second or so to give their answer. So that's pretty significant, 100 milliseconds of difference. Now, we also have good evidence supporting the neural instantiation of a feature matching system in our actual brains. Evidence that this is like indeed how the brain accomplishes recognition at a basic level. Just for example, researchers have identified individual brain cells in our cortex that respond selectively to lines or to curves or to simple shapes. And we actually call these neurons feature detectors. They are a fundamental part of our visual system's neural architecture. Like in the simplest case, early in the primary visual cortex, where the signals go from the eye, we could pick a random neuron and see what makes it fire. And we might find that this one neuron goes off only when we see vertical lines in front of us. Like we flash a horizontal line to the person's eyes and this neuron doesn't give a shit, doesn't get fired up at all. Though some other neuron a little ways away in the brain would. But when we flash a vertical line, this one neuron goes crazy, firing at a super fast rate. Pew, 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 pew. Kind of like a feature demon shouting, me, 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 me. It's a vertical line, it's a vertical line. Meanwhile, if we flash, let's say, an oblique line, right? One that's at an angle, not quite vertical, it might fire a little bit extra, but not a lot, right? Because it's close to being what the feature this one looks for. But the further it is from vertical, the less the neuron fires above baseline. So we've got a neuron that is basically a vertical line detector, a feature detector. And it might be specific, in fact, just to one part of the visual field. Like this one neuron might just be watching for vertical lines in your upper left periphery. And there'd be a different neuron watching for a vertical line in your upper right periphery or upper middle periphery or whatever. And in fact, the way we learned about neurons like this comes from a study, well, a set of studies, but some famous work done by Nobel Prize winning researchers in the 1970s, Hubel and Wiesel. They used an animal model, but we've since replicated all this stuff in humans. But in this case, what they did is they used cats. So they measured individual neurons in the primary visual cortex. So back there at the occipital lobe, in the first real stop-off for processing info from our eyes. They would clamp a little electrode onto a single neuron back there in the cat's brain, one individual cell in the visual cortex, and just measure to see when that neuron fired. Then they had the cat staring forward at a screen without moving its eyes. So the eyes aren't moving at all, which might seem like an impossible task if you ever had a cat, right? But they actually just, they injected something to temporarily turn off the muscles that control eye movement. So the cat actually had no choice but to look straight forward until the injection wore off after the experiment. But when they had the cat fixating at the middle of the screen, not looking anywhere else, they presented different stimuli, just simple stimuli in front of the cat's eyes, like lines or circles, maybe combinations of lines, maybe making up an angle or a corner, maybe some moving lines versus stationary lines, just simple stuff in front of the cat's eyes. And they, because the cat wasn't able to move its eyes to the left or the right, they could present so that they know for sure what they're showing is in the cat's, upper left periphery or bottom middle periphery or whatever. And what they found was that each neuron that they recorded back there in the back of the brain, in the primary visual cortex, each neuron was firing only for a very specific situation in a very specific place in the visual field. Like they would find one cell that only fired for vertical lines in the upper right corner of your periphery, another that only fired for vertical lines in the middle right or the lower right or just to the left of where you're focusing or whatever. But each neuron was just getting a signal from one part of the visual field because it got axons projecting from that light detector at just the right part of the retina, right, where light would always come in at that angle. So things above where you're focusing, things that are in your top periphery, right? That light is going to come into the eye at an angle that'll hit the bottom of your retina, the bottom of your eyeball in the inside, right? So all the wires from the bottom of your retina, they go to one part of your visual cortex, and all the wires from the top of your retina, for things that are in your bottom visual field, they go to a different part of your brain. So it stays organized, and we can have a neuron back there in the cortex that only gets signals from things in the top part of your visual field or the middle left part of your visual field or whatever. And again, they found a lot of these neurons. They found that a lot of these neurons only went off if they saw something very specific, very simple there, generally a straight line or like an edge, basically. They actually called these neurons simple cells because they're individual neurons that respond to something simple, a little line of light at a particular orientation. Like here, we've recorded the firing rate of one little simple cell, one individual neuron in the visual cortex, while we put a line of light in front of the cat's eyes, right? We're just trying out different angles of this little line of light. And when we hit just the right angle, the neuron fires like crazy. And it fires maybe a little bit extra for things that are close to that angle, right? So here's the angle it's looking for, lots of activity. Here's kind of close to that angle, a little bit above baseline, right? And then it fires not at all for things that are a totally different angle. So they mapped out tons of these simple cells all throughout the primary visual cortex. They found thousands of these that actually mapped out every angle of line for every location in the visual field. We have a specialized neuron that went off for just the right input there. Nearby, they also found, just a little further in, they found what we call complex cells. So these are individual neurons that respond to lines of a specific orientation that are moving in a particular direction. So for example, this one specific neuron here only fires when there's a 45-degree bar in the upper right visual field that's moving down and to the right, but not for other angles, or not if it's moving in the opposite direction. And again, we got a ton of these neurons covering every angle for every direction of motion for every part of the visual field. In fact, some of them are okay with the motion going in both directions. Some of them only fire if the motion's going in one specific direction. But we have this vast network of neurons just at the early parts of visual processing in our brain. Vast network of neurons watching for just something simple like this. And not far away, they found what they called hypercomplex cells, although this is still looking for a pretty simple stimulus, a pretty simple feature, right? But they found these hypercomplex cells that were individual neurons that would fire for edges of a particular orientation and length that's moving in a particular direction in a particular part of the visual field. For example, here we've measured the firing rate. You can see just the spikes, how many action potentials there are. So the firing rate of a random neuron that they found in the cortex, and notice how it goes off like crazy for a short upward moving line, if it happens in, let's say the upper left visual field, but not so much for a medium length upward moving line in that part of the visual field, and not much at all for a long upward moving line in that part of the visual field. And of course, it won't fire at all for downward lines or lines at other angles or lines happening anywhere else in the visual field. So we're already starting to build out of simple things like a line. We're getting now combinations, in this case, temporal combinations of lines, moving lines. And indeed, if we go further into your brain, we'll find a neuron that fires only when you see a square shape in, let's say, your right periphery. Like if the white oval here is your whole visual field, what you're looking at, the X is where you're currently looking forward, where you're fixating, whatever's directly ahead of you is the X. Off in your upper right periphery, a square shape shows up. There is a neuron somewhere in your brain that will go off only for that particular combination of features, that combination of four lines with 90-degree angles. Because a combination of four simple cells went off, it makes this square detecting cell go off. So we'd say that this is a new neuron, the square detector. It's tuned to recognize square-shaped light patterns hitting that part of our retina. Meanwhile, somewhere else in that part of your brain is a neuron, or, and I guess in this case, since it's the other visual field, it'd be in the opposite lobe, the opposite hemisphere. But in the other part of your brain, there's a neuron that only goes off for square patterns if they're in your left middle periphery over here. So we'd say that neuron in your brain is tuned to detect square-shaped stimuli hitting the left middle part of your peripheral visual field. It just means that's the only stimulus that makes this one neuron back there in your brain fire. It is selective to squares in that part of the field. But how the hell do we have a neuron in our brain that only goes off for squares in a specific place? How does a single dumb little cell in our brain know what a square is? Well, what is a square except 4 lines at the correct spatial location, right? So imagine we've got those simple cells in our brain that detect individual features. The simplest thing, just a line, an edge, right? If we detect a horizontal edge where this light blue line is, then there's a neuron back in the brain that'll go off. If we detect maybe a horizontal edge a little further down where this green line is, there's a different neuron back in the brain, not far away, that'll go off. If we detect a vertical line far on the left where the red line is drawn, that would make a different neuron in the brain go off. Again, a neuron that's nearby in the brain though. And same for a vertical line in a slightly different location, like the one shown in black here, with its own private neuron that again would be near the others in the brain there. So if a simple line or edge of light hits that part of your retina, its signal ends up going back to a corresponding simple cell in your brain's cortex, right, in the visual cortex of your brain. Now the key is, we could just add one more neuron, one more layer here. We could add a neuron a little further down the line, a little further into the brain, like this one I've drawn in orange here, which might just be wired up to those 4 earlier line detector neurons. That way, if and only if all 4 of them go off at the same time, in other words, if it gets enough combined input from all of them, then this orange neuron goes off. And that would only happen if all 4 lines were there, meaning the orange neuron in your brain will only go off if a square shape actually showed up in that part of your visual field. So the neuron itself doesn't understand anything. The neuron simply is wired up to some other neurons in such a way that it happens to only fire if it gets enough combined input from the 4 other neurons it's attached to. And those 4 neurons, the ones that synapse onto it, they're only going off for a very simple feature there in the visual field. So that's what Hubel and Wiesel discovered and documented, how they got their Nobel Prize, that our visual cortex registers super simple features like lines, just little edges, but from that, other neurons can watch for more complicated combinations of those simple things. So from combinations of lines, we can get squares and triangles. And from combinations of those, we can get houses and other object patterns to recognize. Likewise, our brain can pick up curve patterns, just like, from the right combination of simple cell line detectors going off in the right combination, we got a curve. We can recognize a curve. And with curves and other shapes combined in the right way, suddenly we can start to distinguish faces from houses and from cars and from everything else. All with simple, mindless little neurons just wired up in the right way that they detect something simple, a feature like a little edge. So what Hubel and Wiesel's research with the cats did, it was demonstrate a plausible biological basis for how feature matching might actually work in the brain. Neurons early in the process fire for individual, very simple features, but then neurons later in our brain might fire only for particular combinations of those earlier features. In other words, those neurons will go off only if a combination of earlier neurons goes off. And like I said, that's why they ended up getting the Nobel Prize for this. Now, there's just one problem, because the idea of feature matching, it is widely accepted. It definitely seems to describe how the brain actually works, but it's obviously got to have a lot more complicated stuff going on in addition to that. Like, how do we get from such simple components as lines or combinations of lines to representing or recognizing complex 3-dimensional objects like a lamp or a dog, right? And that leads into another perceptual theory, the one that actually came later, and this new one doesn't undermine or contradict feature matching theory. It actually builds onto it. It's an extension, and this new theory is called recognition by components theory. So recognition by components theory, or RBC theory if you want, was developed by a researcher named Biederman, and it describes how 3D images are identified as recognizable objects. The basic idea here is that our brain breaks down objects into what he called geons, which is just short for geometric ions, just meaning like component parts that are fundamental geometric building blocks, okay? So the idea is that objects that we see and perceive and identify, we're identifying them by their geons, by their building blocks, and the relationship between those geons. Like, our brain recognizes the geons before putting it all together and recognizing the combined pattern as being a dog or a coffee mug. You can see how this really is just an extension of feature matching theory, but into the 3rd dimension. So these geons, these building blocks, they might be things like brick shapes or wedge shapes or cylinder shapes or cone shapes, right? And while the specific details might vary, like some cones are really long and skinny, other cones are short and fat, the idea is that most objects we identify kind of, if they fit into some concept like lamp or briefcase or dog or whatever, they're really just combinations of these geon pieces. Like a mug is really just a cylinder geon combined with a curved handle kind of shaped geon, whereas a briefcase would be a box-shaped geon combined with that curved handle kind of geon, right? So we recognize objects by recognizing combos of those simpler building block pieces. How do we as little children learn to tell the difference between a horse and a giraffe? They've actually got a lot of the same basic parts, right? Basic geons in common. But the giraffe, their combination of geons has longer cylinders at the bottom and longer cylinders kind of hooking the middle up to the top bit. Likewise, recognizing a human shape from an ape shape may start by recognizing the relative lengths or thicknesses of some of the basic cylindrical geons, right? Our limbs. But as bipeds, they're in the same basic layout as us. Likewise for distinguishing different birds or whatever else. Now, obviously there's more than geons involved. We also have systems for recognizing color and texture and other elements that help us distinguish an ape from a human or a spotted giraffe from a non-spotted horse. But when it comes to recognizing three-dimensional objects, recognizing things from shape in a three-dimensional world, the idea in this theory is that our system starts by recognizing component parts, these little geons. Hence, we call it recognition by components theory. The benefit is a theory like this explains how we can recognize the same basic object from different viewpoints and angles. And geons are just made up of edges and curves, right? So as I said before, really this is just an extension of feature detection theory or feature matching theory. An example of empirical support from laboratory experiments. We've used lots of different techniques. There's lots of support for this, but one simple test would just be see how well we can recognize objects when we are able to see the geon information, or how well we can recognize objects when we can't see the geon information, but we get an equal amount of non-geon information. So for example, in one type of task, what we'll do is researchers will black out parts of an object drawing, and in one condition, they block out the geon transition areas, while in another, we might actually be able to see the geon transitions. And we find that people are much better in the latter condition. So for example, what's the object in this picture? Take a moment. There's something behind all that black, right? But most people have trouble figuring out what it is from what we've got here. It's kind of hard to see. Most people can't get this. But now look at the top version. So it's the same object, but when we can see the geon information, most people quickly recognize what it is. And they're like, oh, it's a flashlight, right? It's got the little button on the top. It's a flashlight. But we can only see that because we can see the geon information. So that tells us that that is important information for recognition. And just like with feature detection theory, we've actually got quite a bit of support on the neuroscience side, suggesting that the brain is indeed doing something like this. So for example, we have discovered that some neurons, individual neurons, selectively respond to what we call viewpoint invariant properties of an object. For example, there's a neuron that fires specifically for the shapes that a cylinder can take, the different shapes, but nothing else. Like regardless of angle, it fires for a cylinder, different versions of a cylinder. So it seems to be like a cylinder geon detector. And we've found neurons like that in the brain, for example. But there is a problem, or at least a limit of this. Recognition by components theory doesn't really explain how we make fine-grained distinctions, like distinguishing between a friend's face versus a stranger's face when both faces are made up of the same basic geons, right? So there's gotta also be some extra pattern recognition built in that focuses on smaller scale features and their combinations. But the basic idea of recognizing complex things from combinations of simpler features, recognizing a whole by the parts that make that up, that is well established. That is a fundamental part of how the brain recognizes things visually. We'll actually stop here for the theory video, but one thing I'll note is that the theories we've been talking about so far, those are bottom-up theories. They're looking at how the system can make sense of things purely based on interpreting the input as it came in. In other words, a straightforward analysis of the proximal stimulus hitting our retina. But we already saw lots of ways in which the brain doesn't rely purely on what's in the proximal stimulus. On this bottom-up analysis of the information. Like that's not, our brain does more than that, right? It also takes into account contextual cues, our knowledge, what else we may have seen recently that predicts what we might be looking at, all sorts of other factors that come from stuff already up there in our brain, not from the stimulus itself. So we call those factors top-down factors, and that's actually what the next video will dive into.